my name is Kristin Jones. My partner Julianna Procaccini and I will be talking to you about the enzyme Penicillin G acylase. Penicillin G acylase is the recommended name of our enzyme, however it is also known as PGA. It has common names such as Penicillin G amidase and Penicillin G amidohydrolase. It can be found in UniProt under the gene name PAC, and it is also found in the protein databank under the ID 1FXV. PGA is naturally found in E. coli, however it can also be found in yeast and fungi. Within these organisms, it is found within the periplasm, which in bacteria is found between the cytoplasmic membrane and the outer bacterial membrane, and in yeast and fungi is in between the plasma membrane and the cell wall. Correct folding and processing of PGA does not occur until it reaches the periplasm, and this is to ensure that catalytic activity does not start occurring until it is in the correct location within the cell. The function of PGA is to produce 6-amino-penicillinate, which is the starting material of the production of semi-synthetic penicillins, such as ampicillin, carbenicillin, and oxacillin. And PGA is so amazing for its function because being college students and being around other people's germs and being sick all the time and frequently needing antibiotics, without enzymes like PGA, we wouldn't be as available to antibiotics and they wouldn't be as mass-produced. The in vivo role of PGA is unclear, however, it is thought to be involved in the assimilation of carbon sources for the living organism. The enzyme classification for PGA can be found in the protein databank. It indicates that it is a hydrolase, meaning that it catalyzes the breaking of bonds using water. And specifically, it hydrolyzes the carbon to nitrogen bonds within linear amides. The enzyme classification 3.5.1.11 is our enzyme penicillin acylase. So this is the primary sequence of PGA. The entire enzyme consists of 846 amino acid residues, and it is divided into 4 segments. An N-terminal signal sequence, 2 polypeptide chains, and a spacer peptide between the 2 chains. In green here you can see the signal sequence, the N-terminal signal sequence, which is used to direct the enzyme to the periplasm. Chain A, shown in red, is 209 amino acids long. The spacer peptide in between, here in yellow, is used to ensure correct folding of the enzyme. And chain B, shown in blue, is 557 amino acid residues long. So this is a secondary structure map of chain A, and basically this just shows the residues that are that fold to form the secondary structures, and it's used to locate the alpha helices and beta strands and beta turns, and it also shows the amino acid residues that are involved in ligand contact and also the metal cofactor contact, shown in these red dots and blue dots. This is the secondary structure for chain B, and it shows the exact same thing. It shows the alpha helices and the beta strands and beta turns, and it also shows the residues that are involved in ligand binding and the metal cofactor binding. This is the tertiary structure of PGA, and it is characterized by 2 polypeptide chains that are divided into 5 domains. Chain A is divided into 2 domains and is shown here in blue. And chain B is divided into 3 domains and shown here in red. The 2 polypeptide chains are tightly entwined and they form a deep cup-shaped depression within the center, and within this center is the hydrophobic binding pocket. And aromatic residues line this binding pocket, and they are major contributors to substrate recognition and binding. PGA has a very broad substrate specificity. It does primarily bind to penicillin G, but it can bind to basically any linear amide, and as long as it's a R to CO to NH to R prime, and as long as the R is hydrophobic. It also binds to a calcium metal cofactor. The catalytic site of PGA is a single amino acid catalytic center, which is located in the mouth of the hydrophobic binding pocket. And this single amino acid is serine 290, and it is also known as serine B1 because it is the first amino acid residue in the B chain. So the mechanism of PGA is characterized by penicillin in water forming a carboxylate in a 6-amino acid or a 6-amino penicillinate. Here you can see the penicillin in the exact cleavage site of the amide, and this is the product, the 6-amino penicillinate. The catalytic mechanism of PGA is very similar to serine proteases in that it uses a serine as a nucleophile. However, it doesn't utilize a catalytic triad and uses water to deprotonate the nucleophile. The catalytic mechanism begins by deprotonating the serine, which goes on to attack the carbonyl carbon of penicillin. A tetrahedral intermediate is formed that is stabilized by hydrogens, and the electron transfer from the oxygen occurs and the release of the N to R prime, which is the 6-amino penicillinate product. The second part of the mechanism begins by water acting as the nucleophile, which attacks that same carbonyl carbon, and another tetrahedral intermediate is formed and stabilized by hydrogens within the main chain. Another electron transfer occurs from the oxygen and a carboxylate product is released, and the enzyme is regenerated. My partner will now discuss the primary literature paper on our enzyme.
Hi, I'm Julianna, and I'm going to be talking about our primary literature paper. For this part of the project, we chose the effect of pH in the synthesis of ampicillin by penicillin acylase. So the background of this paper is that it was written and published in 1996 by Enzyme and Microbiotechnology. The study was done in Mexico and written by the following authors on the screen. So for our introduction, beta-lactam antibiotics include both penicillin and ampicillin, and one of the structures that's well known to these is that they all have a beta-lactam ring, which is a 4-membered ring that has nitrogen on one of the sides. Ampicillin is, it inactivates the penicillin bind proteins on the inner membrane of bacterial cells, and it's really common antibiotic for pneumonia, urinary tract infections, staph infections. It has a wide range of uses. It also has a wide specificity, and it binds to multiple substrates. And 2 of those are PGME and 6-APA. PGME is phenylglycine methyl ester, and 6-APA is 6-amino penicillic acid. So what the problem is that pH directly affects how much ampicillin you produce. Different pH levels will give you a different amount of ampicillin. So the study was done to see what the optimal pH level would be so they could maximize their production of ampicillin. And this is important because being able to find this optimal level will allow you to maximize your production, drive down costs, and also be able to distribute antibiotics in a wider spectrum. So for their methods, they used recombinant E. coli and placed it in agar plates, and all of these plates were then placed in a stir bath reactor, and this reactor was at a constant temperature of 25 degrees Celsius. They never changed the temperature for any of their 3 experiments. Before actually starting all of this, they used Lineweaver-Burke plots to be able to get the Vmax and Km values of PGME and 6-APA to be able to use this in their later studies. And this was, different pH levels were tested in the batch reactor by changing the amount of 2 molar ammonium hydroxide in the reactor. And the reactor also had 1 molar potassium phosphate in it as well. So for their results, they wanted to test the relative activity of enzyme at different pH levels. Figure 1 was their first experiment. So what they did was they compared PGME hydrolysis to different pH levels, and this shows that at a pH of 7, which is this top point right here, this was a high rate of hydrolysis, which meant it was a very slow reaction at a pH of 7. Well, at this pH of 6 on this linear portion, it remained constant through hydrolysis, which improved its synthesis by 53%. For their figure 2, which you can see here, this was their second experiment, they compared the initial rate of synthesis of ampicillin with pH levels. At the pH of 6, which is the top point right here, it was 1.3 times faster rate of initial synthesis compared to that of the pH of 7, which is the lowest point here. And of note, they didn't do any studies down in this lower range because they assumed that this would be a decrease in the amount of synthesis because of the free protonated enzyme in this area. Their third experiment that they did is they tested the pH of 6 and the pH of 7 with PGME and 6-APA. They found out, due to the wide specificity of this enzyme, it was able to bind to both PGME and 6-APA. At the pH of 6, both PGME and 6-APA produced 17.45 more milligrams of ampicillin per unit of enzyme per activity of the batch, compared to that of at the pH of 7. They also did this study and were able to determine that substrate dissociation is independent of the enzyme complex, and they also found out that it's non-competitive inhibition in E. coli. So for their conclusions, they found out that pH indeed can change the rate of synthesis of ampicillin. Back to figure 1, it showed that at a pH of 7, hydrolysis was high, so this was a slower reaction, which meant since it was a slower reaction, you're producing less ampicillin than you are at a pH of 6. And for figure 2, they showed that since the initial rate of synthesis was higher at a pH of 6 than at a pH of 7, it was able to produce more, and it was more efficient at the beginning of synthesis. And when they did a larger study, they found out that 75% more ampicillin was made at a pH of 6 than at a pH of 7. And they also found that for the reaction mechanism, it was a second-order reaction, but it was not a ping-pong reaction. They actually assumed at the beginning of their study that it would be ping-pong, and all the steps would happen simultaneously, but they found that the methanol, not simultaneously, and they found that the methanol released by the enzyme happened before the amide linkage with the enzyme intermediate. So they all happened simultaneously, but they expected that they weren't going to happen simultaneously. So this is important because ampicillin and penicillin are main antibiotics for treating infections such as strep throat and staph. And without ampicillin, strep throat is fatal, so this antibiotic is important. And the synthesis of ampicillin cannot happen without enzymes such as PGA, so it's of wide importance to us. And also, this study was important to be able to produce these optimal values of ampicillin. If you're going to produce something at 4 pH and you're getting less, but you're going to get more at a pH of 6, you should go with the pH of 6 to maximize your production. So what's next for this study? I thought that they'd be able to test other antibiotics such as Keflax, Bactrim, Cipro, and see if those had optimal pHs to maximize their efficiency. And I think studying to see why it's not a ping-pong reaction and why those steps all happen simultaneously would be interesting. Research on temperature denaturation would be also interesting because they tested all their experiments at 25 degrees Celsius, but maybe changing that to 25, 26 degrees Celsius or 24 could also be more optimal to produce ampicillin. And I think it's interesting why PGA has such a wide specificity instead of a high specificity for one substrate. So these are our primary sources that we used for our paper, as you can see on the screen. Then these are our website citations that we also used in our project. And finally, these are our image citations for all the images we used in our PowerPoint presentation. And that's the end of our presentation. Thank you.